Chaz is assistant professor of medicine in the Division of Infectious Diseases at UC Health. He's a scientist at the Chaz Langelier Biohub. He's also the associate medical director for hospital epidemiology and infection prevention, certainly an important role at this time. And we look forward to his talk about diagnostics.
Great, thanks very much for the kind introduction, David. I appreciate the opportunity to talk about COVID-19 diagnostics, some of the great team science that's being done in this area at UCSF and at the Biohub. You know, I just want to start by saying that diagnostic testing has really been a foundational piece of the COVID-19 response. So important for understanding community prevalence and so important for preventing infection transmission in the hospital. All you need to do is take a look at the headlines or some of the commentaries in leading journals, and you recognize that diagnostics have been a hot issue in the United States. Next slide, please. Next slide, please. And if you take a look at the per capita, if you could go back, please. Just taking a look at the per capita testing in the United States kind of highlights one of the key issues, and that's diagnostic test availability. The US doesn't stack up so well against many of the other countries that have been faced with this pandemic. That was due to multifactorial reasons, but one good thing that helped to change this was on February 29th, the FDA revised their guidance and allowed for individual laboratories such as the UCSF Clinical Microbiology Laboratory to develop their own diagnostic tests. Most of these were reverse transcriptase PCR tests. And I want to give a shout out to Steve Miller, Ed Thornborough, Charles Chu, and the Clinical Microbiology Lab, who within just a week were able to validate a reverse transcriptase PCR test for UCSF. And that was really helpful. It provided testing availability, mostly to UCSF inpatients, but wasn't able to meet the need for all of the UCSF inpatients, not to mention outpatients. And major institutions like Zuckerberg San Francisco General, like San Francisco Department of Public Health, and many other areas still did not have availability for diagnostic testing. Next slide, please. And really, in one of the more inspiring, if not most inspiring examples of teamwork for a common cause, in little under a week, over 50 volunteers, scientists, and other individuals came together to build an extension of the UCSF Microbiology CLIA lab in the space below the Chaz Langelier Biohub on the Mission Bay campus. And leveraging the advanced analytics, liquid handling robots, and technology, as well as volunteer scientists, this CLIA lab extension was built. Next slide, please. And you know, this spanned from equipment and supply chain management to robotics to high throughput PCR to compliance and validation, and really has notably expanded the testing capacity both at UCSF and allowed for testing at other institutions. Next slide, please. So this is a research talk, and so it's important to talk about research. I think one can't talk about diagnostic testing coronavirus research, however, without reflecting on the past and without reflecting on the technology that led to the discovery of the original SARS coronavirus. In 2003, the ViroChip was about as cool as it gets. This was built by Joe DeRisi and provided an ability on a microarray type format for detecting all known viruses at the time. Things have changed since then. In 2020, what was used to discover the SARS coronavirus 2, the cause of COVID-19, was a technique known as metagenomic next generation sequencing. You can see one of the most high throughput sequencers there on the right. Next slide, please. And one of the key aspects of metagenomic next-gen sequencing is that it's unbiased in terms of its ability to detect pathogens and its ability to survey the world. Next slide, please. And it's unbiased because simply it requires detection of nucleic acid that's present in all biological entities. And this allows for pathogen discovery in a way that other diagnostic tests can't. Next slide, please. So let's move on to some data. I want to show a snapshot of a pilot study looking at 30 of UCSF's COVID-19 patient under investigation, PUI, suspects. We can take a look here, and what you're seeing is a heat map of reads per million of sequences aligned against different viruses. And you can see that 21 of these samples had alignments to SARS coronavirus 2. You can also see that in a number of the unknown cases, other respiratory viruses were identified, including RSV, and one of the common coronaviruses that actually isn't detectable by many hospital PCR panels, coronavirus NL63. Next slide, please. And not only was a detection allowed with this technology, but in many cases, entire viral genomes were recovered. You can see that the coverage breadth here was 100% across the SARS-CoV-2 genome at a depth of over 6,000. And this allowed for unprecedented analysis of single nucleotide polymorphisms that can provide very informative data. Next slide, please. One of the key things that one can do with this type of data is understand genetic relatedness across SARS coronaviruses, to construct transmission maps, to identify novel mutations. In fact, 7 new polymorphisms or mutations were identified just in this initial data set. And you can appreciate the relatedness of those samples from UCSF, shown in blue, to other coronavirus genomes from other parts of the world. Next slide, please. I want to give a shout out to a really amazing metagenomic sequencing and data science team that has come together to make this possible at the BioHub. Really another fantastic example of the teamwork that is in place that is aiming to not only do metagenomic sequencing, but to collect all of the genomes from SARS coronavirus 2s that are identified at UCSF and at many sites in Northern California, make these publicly available, make them available to allow for genomic epidemiology studies, to understand transmission throughout the world, to understand potential genetic relatedness between different viral strains and outcomes. Next slide, please. When I was talking about metagenomic sequencing, I noted that it was unbiased in terms of being able to detect nucleic acid from any biological entity. And one of the key entities is the host. As we learn more about respiratory infections, we recognize that it's not just about the virus, it's not just about a pathogen, but really it's a dynamic interplay between pathogen, host, and in many cases, microbiome. Next slide, please. And this is important because we can learn a lot from host response, from looking at host gene expression. For instance, on the left, I'm showing a heat map looking at expression of genes that are most different between patients with pneumonia and patients with severe respiratory illnesses that appear similar to pneumonia, but ultimately are non-infectious. And we can see that by looking at host gene expression, one can differentiate those. A fantastic computational biologist named Katrina Kalantar worked with our group to use a machine learning approach to build a 12-gene classifier that could distinguish patients with true pneumonia from those with severe non-infectious acute respiratory illnesses. And this performed with very good accuracy. You can see the receiver operating characteristic curve on the right. Next slide, please. This has a lot of relevance in terms of COVID-19 because one of the most severe outcomes, one of the most feared outcomes that happens in a subset of patients is the acute respiratory distress syndrome, which can lead to persistent hypoxemic respiratory failure and in many cases progress to mortality. I want to show work here from Arctic Sarma, Stephanie Christensen, and Carolyn Calfee, from whom you'll hear in just a bit. And this is a heat map looking at gene expression differences in patients with ARDS versus those without in UCSF critically ill patients. It highlights heterogeneity, but it also highlights the potential to identify these subgroups of patients who progress to severe disease. And one thing that we're hoping to do is to use this type of transcriptional profiling approach, transcriptional data that one obtains from metagenomic sequencing to be able to predict outcomes. Just like in that pneumonia classifier, we hope to be able to predict who is at risk for progressing to ARDS, who is at risk for having severe outcomes in COVID-19 patients. Next slide, please. So I've just highlighted some of the genomic work that is ongoing on the diagnostics front. And genomic diagnostics have a lot of potential to answer important questions during acute disease. But really, one of the key unmet needs in the diagnostic realm is understanding who's been exposed, who has made antibodies against this SARS coronavirus, and who potentially is immune and can work and participate again. So with that, I'm going to turn things over to Colin to talk about serologic diagnostic assays and some very cool stuff under development.
Hey, everybody. Yes, my name is Colin Zamanek. I'm a scientist in Joe DeRisi's lab, as well as across in neurology and Michael Wilson's lab. Thanks a lot, Chaz. I just wanted to talk briefly about some of our efforts here on highly parallelized serology discovery and development and why those are important. So today, all of our diagnostic efforts have really focused on direct detection, and that's mostly done by qPCR. So this looks for viral DNA in the sample, usually from a swab in the nose or throat, and is typically very sensitive. However, these tests are really limited by the fact that there has to be live virus in order to kind of be able to detect whether or not there's DNA there. There has to be virus there, right? And so therefore, the detection window is pretty short. Serological profiling, on the other hand, or antibody diagnostics lets us look for antibodies from our own immune systems that they generate against the virus. So based on the kind of antibodies that we generate, we can distinguish between acute and past infection. So this is critical because it gives us insight into who is exposed and, most importantly, who can get back to work. So unfortunately, these are really slow to spool up. So once they're made, they're very easy to scale. Next slide. So why is that? The biggest challenge is that we don't know what part of the virus the antibodies target. The typical workflow sort of starts by generating standalone proteins. You may have heard about spike or capsid proteins that are present on these coronaviruses. And then we'll sort of pan those against known positive patients and then test whether or not binding is specific to this virus and excluding it as a potential target if there's cross-reactivity to other coronaviruses that might be out in the wild today. So this is pretty tedious and slow and limited oftentimes by protein production efforts. Next slide. So our approach is to massively parallelize this discovery effort using phage display. So in a nutshell, we test all of the possible viral protein components at the same time. So we first synthesize all of the pieces of the viral genome into 38 amino acid chunks and then clone those into the backbone of a T7 bacteriophage system. This makes a library that has all of the protein components of the novel coronavirus that are displayed in pieces on the surface of respective phage. And that lets us simultaneously pan antibodies from patients that have coronavirus against all the possible antibody targets at once. There's several critical outcomes from this. One is that we can identify common biomarkers in an unbiased way from patients that were infected and rule out ones that were cross-reactive from other viral infections. So we can include all kinds of other coronaviruses and other viruses in these libraries, which in fact we do. We can also deeply characterize the adaptive immune response in an unbiased way. And downstream, this lets us track patients who did well and those who did not, and ask whether their antibody targets were different. This helps us inform better vaccine development. Next slide. Lastly, we can use these phages that express viral peptides for use in serological diagnostics themselves. Our lab first uses the whole library approach to identify some critical key candidates and then break those out of the pool and validate them one by one. So then we can grow up that particular phage clone and use it in a test strip or a bead-based assay, which avoids many of the challenges of typical recombinant protein production. We're super excited to be working alongside a broader serology effort with Chaz and other infectious disease efforts at UCSF to help answer questions like who can get back to work and who's already immune to the virus. And this has been an incredible group effort from several labs, particularly the Weill Institute for fronting major funds in order to get this effort up and running very, very quickly. My colleague Jay is Jay Rajan, who's an infectious disease expert here at UCSF, as well as in Joe DeRisi's lab, who is an expert in viral phage display, as well as Michael Wilson and Joe, who are really also experts in this space. Chaz and Carolyn Calfee, who are working with us on getting the critical patient samples in order to validate these assays, and the BioHub for producing some test proteins and working with us on a large scale. Thanks, everyone.